Hi, how are you? I hope you're doing well. Let's get started into solving May 2025 Unit 3 Practical Skills in Biology. For the first question, we've got a very direct question. State the 4 stages of mitosis in the order in which they occur. And we have memorized this before using the mnemonic P, mat, prophase, metaphase, anaphase, telophase. Just write them in the correct order in this question. Let's read it as well for the rest of the questions. Trifluralin is a chemical used to stop the growth of weeds in fields of crops. Mitosis is a process that is essential for the growth of weeds. The effect of trifluralin on mitosis on wheat plants was investigated. We've got wheat grains that were germinated on moist filter paper. The wheat seedlings were transferred to Petri dishes containing discs of filter paper as shown in the photograph. The filter paper discs were soaked in a solvent or the solvent containing 7 mg centimeter cube of trifluralin. The Petri dishes containing the seedlings were incubated at 25 degrees Celsius. For the first question, we're gonna get asked why these Petri dishes contain seedlings incubated at 25 degrees Celsius, or why have we actually used 25 degrees Celsius? So whenever we get asked a question about why this temperature was used, this is definitely to be optimum for the enzymes in this investigation. For the enzymes in mitosis, I need it to be 25 for a reason. I don't want it to be higher than that, because if it was higher, the enzymes will denature. So I don't need it to be too high. It's not too high to prevent enzymes from denaturing. And I don't want it to be less than 25 degrees Celsius, for example 10. I do not want it to be 10 degrees Celsius, because this will make the enzymes work slowly. So it's something in the in-between of the maximum number that will make the enzymes denature and the minimum number that will make them work inactively. I just need it to be optimum for the enzymes. State the independent variable in this investigation. While the independent variable is the changing variable, or the variable that was changing in this investigation, and the effect that we are investigating. The effect here of trifluralin on mitosis. So the independent variable is the presence of trifluralin. And that's going to be my answer. Presence of trifluralin is the variable that I'm investigating its effect. Give 2 reasons why the grains were germinated before being transferred to the Petri dishes. We have germinated these grains before being transferred. Of course, I need them to be viable. I need them to work. I need to make sure that they all started the treatment at the same stage. They all started the treatment when they were germinated. So that's going to be basically my answer, that I need to make sure that the seeds were viable in this investigation, and to ensure they all started the treatment at the same stage where they were all germinated. None of the seeds were not germinated, and none of them were in a higher stage of development. They were all starting at the same stage. So that's why we have germinated all of them before being transferred. Explain why some of the seedlings were placed without trifluralin. And that's basically whenever we get an experiment with the same everything, the same conditions of everything, but without the independent variable being investigated. This is mainly to act as a control most of the time, and we're gonna elaborate why we use a control in the first place. We use a control experiment to eliminate the effect of other factors. I need to remove the effect of other factors than the presence of trifluralin. So I need to only get the difference between the results having the control experiment and the experiment having trifluralin, to make sure that the results have the difference, and that difference was due to the independent variable that we used. So that any changes using trifluralin could be attributed to trifluralin only. So any changes due to any other factors in this investigation other than the effect that we are investigating will be removed. I only need the effect of the independent variable that we are using. So now, after 12 hours of incubation, root samples were taken from seedlings in each Petri dish. We have taken root samples. The root samples were examined to determine which stage of mitosis were present and how many cells were in each stage. The bar chart shows the percentage of cells in each stage of mitosis numbered. We have the stages numbered, of course, not in names, because we were already asked about their names in the first question. So I have stage 1, stage 2, 3, and 4. And we have data with the solvent without trifluorelin and the solvent with trifluorelin. Alright, we can see here that we are seeing the comparison with the control experiment, so that the difference between them, of course, is the effect of trifluorelin alone. And that was the answer to the question that asked us, why have we used a control experiment in the first place? So now let's see the question. This is the core practical of the root tip squash. Describe how a root sample could have been prepared and examined to obtain these results. So actually we get a root tip. We cut the last 5 millimeters of the part which is the meristem. The root tip meristem is the part where the most active cells are present in mitosis. So we cut the last 5 millimeters, or we could say 4 millimeters from this root tip, and we cut them. We prepare them to be examined for mitosis. We're going to describe all the stages here because this is what the question is asking us in this question, in order to get this view of the cells, in order to be able to see the cells in mitosis. For example, this is in metaphase, and we have these cells are all stained with acetic orcine, or whatever stain you could say. You could say methylene blue, but this one is red, so we could say this is acetic orcine. Of course, these pages are not in the exam. I just added them for clarification. For the points that we have to write to get the full mark in this question, we need to obtain a root tip and cut the last 5 millimeters of the root. We have to place it in a warm HCl, hydrochloric acid. We have to place it in an acid, of course, to separate the cells. It has to be warm as well. Place it on a microscopic slide and in acetic orcine stain. Acetic orcine stain is the part that's showing this reddish color. If we didn't have a stain, we couldn't see the cells clearly, or we couldn't identify the cells without a stain anyway, so we have to use this stain. Heat to intensify the stain. Tease the cells apart using a needle. Teasing the cells occurs where we get a needle and insert it in between the microscopic slide and the cover slip in order to separate the cells and see them clearer. Apply pressure by squashing gently the cells underneath the cover slip. We could apply pressure using our finger by applying pressure here on top of it, the cover slip. Firstly, in order to view this using a microscope, we have to mention that we need to view on low power first to locate the cells, then high power to magnify. This point wasn't mentioned in the marking scheme of this exam, but it was actually mentioned in other previous exams, so we have to add both points to guarantee the marks. For the marking scheme, view on low power first, then locate the cells, and high power to magnify. We need to count the mitotic stages and, of course, each cell in each mitotic stage. Next question, it says that there were 98 cells in total seen in the samples from the seedlings in the solvent with trifluorelin. Calculate how many cells were in stage 2 in the samples from the seedlings in the solvent with trifluorelin. So first of all, I have to see what stage 2 was in the solvent with trifluorelin. I have to see the data in the graph here with the solvent with trifluorelin, which means it's the darker one for stage 2. The darker bar chart for stage 2 is at 80%. So 80% of these cells are the cells in mitosis. So I have to multiply 98 by 80% to get the final answer. This will be 78.4 using your calculator, but make sure to not write 78.4 here because we do not have a quarter of a cell. We do not have a fraction of a cell, so you have to make it 78 in order to be a whole number. So my answer here will be 78 cells. Explain the effect that trifluorelin could have on the processes in mitosis. So let's see the table in order to explain these effects. Trifluorelin actually had no results starting from stage number 3. We had totally zero number of cells in mitosis for stage 3 and also for stage 4, which means that mitosis has stopped after stage 2, which was metaphase, and starting from anaphase, no mitosis was seen. There were no cells in anaphase, stage 3, and of course in telophase 2. We could give a reason for that. The trifluralin has stopped the spindle apparatus from forming. There was no spindle fibers and the chromosomes were not moving apart. We know, of course, that in anaphase that the cells move apart to each side of the cell poles. So if these chromosomes were not pulled apart to each pole of the cell, this means that there were no spindle fibers formed to pull them apart. We had no spindle fibers. And this is going to be my explanation. Take care that explain questions need you to describe from the data to show how you got this information, and then follow this by some explanation from your own knowledge or from the syllabus. We have deduced here that there are no cells in anaphase. That's why we could say that the chromosomes were not moving apart and the spindle apparatus is not formed. Moving on to the next question. Some alcohols such as methanol increase the permeability of cell membranes. Cholesterol can reverse this effect. This is already a piece of information that we already know in our syllabus. Membrane permeability in the presence of these chemicals was investigated. Beetroot cells contain a red pigment called betalain. Membrane permeability of the beetroot cells was estimated by measuring the loss of betalain from slices of beetroot. So this also was a previous core practical in your Unit 3 syllabus, that we measure the loss of betalain from slices of beetroot in order to investigate the membrane permeability. Samples of beetroot were immersed in four liquids: methanol only, methanol with this concentration of cholesterol, methanol with a higher concentration of cholesterol, and water. Water, of course, is a control experiment in here. Samples of each solution were taken every 5 hours, so this is my time interval. Membrane permeability was estimated from these samples, and we have a graph that shows the results of this investigation. And the graph, of course, I have every treatment here found on the right side, and membrane permeability is here, and this is the time. Justify each step of a method that could be used to obtain the results shown in the graph. So basically, in this question that's asking us to justify, I need to state my steps that we already use. I'm gonna say the steps of this experiment as we have already learned before, and add to this why we did that. So if I did every single step, I have to say, why have I done this? Or what's the justification behind this? So we could look at a sample answer following the marking scheme. We could write that. Firstly, in order to start any experiment, I have to say, what's my sample? What am I going to use? Get equal sized pieces of beetroot cut. Alright, they are equal sized pieces, so I have to reason this. Why do I need them to be equal? Because we need the size to not affect the quantity of the pigment. If I got greater pieces of beetroots, this will affect the quantity of the pigment. We're going to have a higher concentration and a higher volume of pigment present inside these beetroot cells, so this is not going to be a valid experiment. Secondly, we have to rinse the beetroot pieces under running water to rinse them, because after the cutting we're going to have some stains of this pigment excessively leaked outside, and this is not by the act of permeability, it's not by the act of leakage, it's by physical damage. It's by cutting using, for example, a knife or something, and this knife has caused a physical damage, causing some leakage, or we could say some excess pigment leaked out by physical damage. So we rinse that to remove any excess pigment. For any experiment, we also need some controlled variables. We make sure temperature is kept constant. Temperature affects any experiment, and for this experiment, it can affect the pigment leakage. So I have to reason this. I have written all the reasons in pink, so you could make sure to include them, because it can affect the pigment leakage Secondly, we have to remove the beetroot pieces from the solution. Of course, I need to remove them in order to measure the color intensity. I don't need to keep the beetroot in its position. It has to be removed from this whatever container it was inside. We remove the beetroot pieces from the solution. Then we have to shake the solution. So if I have, for example, a test tube, I have to shake this test tube in order for the pigment inside to diffuse or to get dispersed equally in the container. It has to be dispersed to have a homogeneous concentration or a homogeneous light intensity of this pigment. Use a colorimeter to assess the quantity of the pigment lost. I'm going to use a colorimeter to measure the color intensity of the pigment lost inside this test tube. We could also say for the reasoning that the colorimeter is a quantitative method, and we could also say this is another option to write to gain the same mark. We could say that we have to zero with the plain solvent in the cuvette, and this is to remove any systematic errors. Using water, as we have already said a few minutes ago, that using water acts as a control. Repeat in order to check reliability and calculate mean values. So the reasoning for repeating is in order to check reliability. And that's it, guys, for this question. I hope you knew how to get the 5 marks fully without losing a single mark. Draw a table that includes only the results obtained at 20 hours and take care from such idea it needs us to include only the results obtained at 20 hours. So guys, please don't include everything, because I've seen answers to this table that have included 5, 10, and 15, and we don't need this. We only need the results at 20, so I don't have to include the time in my axis. I don't need to do that. I only need to get the treatments on the x-axis and the membrane permeability on the y-axis, or we could say for a table this would be on the left side of the table and the membrane permeability is on the right side of the table. And let's write this to see how we could do it. So the treatment here has to be on the left side of the table, membrane permeability at 20 hours, and don't forget the unit of arbitrary units. And we have to get these values from the table and write them from the graph and write them correctly in the table. Plot a suitable graph for the results obtained at 20 hours. Now I know that we have already got a graph here, but we only need a graph for the 20 data alone, not including the 5, 10, and 15 times. So if we have to plot this, let's firstly make sure we include the labelings of the axis correctly. For the x-axis, we need to write treatment or the solutions used. And for the y-axis, we have to say membrane permeability at 20 hours. So these will be my labels of the axis. Of course, we're going to use bar charts. As we have names of the treatment, they're not numerical, so I have to include names of these using a bar chart. So as you can see, I have placed the control methanol plus cholesterol for the micro and the milli units. For the y-axis, I have the minimum value at 0.8 and the maximum value at 4.7. So my scale could be using a scale of 1, increasing by 1, 2, 3, 4, 5. Yes, that would be suitable. So I could use 1 here, 2, 3, 4, and 5. Then place your bar charts accurately according to this scale. And here you go. This is how I would place my bar charts. Now let's see this question. The diagrams show parts of membranes around beetroot cells soon after the experiment started. We have diagram A with a membrane with methanol only and diagram B containing cholesterol as well. Alright, we have these are obviously the pigment particles or the pigment molecules. This is a phospholipid, and the ones embedded inside the membrane here are the cholesterol molecules. We have a key here that this is the betylene molecule, or the pigment, which is the betylene. We have this, which we have already said that this is the cholesterol. So just right here, complete the key by naming the molecules. Cholesterol is here. And this is obviously the phospholipid. Complete diagram A by labeling the inside and the outside of the cell. Alright, the inside and the outside, this has an idea in diagram A. For diagram A, I can't really know what is the inside and what is the outside. But I can take a look at diagram B, which has a well-fitted permeability, or like the correct permeability that a membrane should have containing cholesterol. And we could say here that this side has these pigment molecules. So this side is the inside of the membrane without leakage. I don't have leakage here. I only have these inside the membrane. So this is my inside of the membrane, and this is the outside of the membrane where the leakage can occur this way. Alright, draw an arrow next to diagram A to show the net movement of betylene during the experiment. And we have already said that they move from inside to outside. This is leakage of the betylene molecules. And we have noted that from this diagram that this is the inside and this is the outside. Now let's move to the next question. Cassava is an important crop because of the high starch content of the roots. The value of the crop depends on the starch content, so this must be assessed accurately. The photograph shows a whole and a sliced cassava root. Describe the structure of starch. This is a direct question from Unit 1. We could say that starch is a polysaccharide made from a mixture of amylose and amylopectin with a monomer of alpha glucose molecules joined by glycosidic bonds. I'm highlighting the keywords in the marking scheme, by the way. We could also elaborate using information about amylose and amylopectin separately. We could say that amylose is joined by 1,4 glycosidic bonds only. However, amylopectin is joined by both 1,4 and 1,6 glycosidic bonds. Explain how color standards can be used as a semi-quantitative method of estimating the concentration of starch in a sample. So for this question, we're gonna definitely get the sample first with known concentrations of starch. We're gonna prepare known concentrations of starch and add iodine solution to each sample. This will be the result using a range of concentrations of starch and adding the same concentration of iodine to each of them. We're gonna get the results here, knowing that this one has the lowest concentration of starch, this is a bit higher, and this is a bit higher, and this would be the highest ever of all of them. Why is that? It's due to the color we have here. This is the lightest color and this is the darkest color. As the color darkness increases, this indicates more starch present. Right, we could say that we compare the color of the sample with added iodine to a color chart. We could get a color chart with known standards, and we should say that the darker the color, the more the starch, or the reverse. With the reverse is, of course, the lighter the color, the less starch present. So this has the least starch and this has the highest starch. Describe the difference between a result obtained from a quantitative test and a result obtained from a semi-quantitative test. This question was pretty simple as well. We should only say that the quantitative result is exact, while the semi-quantitative result is an estimate, or we could say it involves an element of judgment, and an approximate, a subjective value. All of these are correct. The starch content of 9 regions of a cassava root was investigated. The root was cut into 3 sections, A, B, and C. Each of these sections has an outer, middle, and an inner layer. The 9 regions are shown in the diagram and the table. The starch content was measured in each region in grams per 100 grams of tissue. The table shows the results of this investigation. We have the layers of section, outer, middle, and inner. A, B, and C, all of them are per gram per 100 grams of tissue. The question says calculate the percentage difference between the highest and lowest values obtained from these 9 regions. So firstly, I have to identify the highest and the lowest values. My highest value of all of these should be 33.65. There's nothing higher than this in these values. The lowest value is also, we could say, yes, 29.64. So these are the highest and the lowest value. I just need to get the percentage difference, but take care, it's using this formula, difference between 2 values. So I have to get the difference between 33.65 minus 29.64. But that's not all. I have to get the mean of these 2 values. So you could get the mean of these 2 values. The mean of any 2 values is their sum divided by their number divided by 2 here. So the mean value will be 31.65. So just write this difference divided by the mean value of both of these values, 31.65 times 100. The final answer should be 12.67. We're not going to round it up into 12.7 or any of these values. It's also accepted in the marking scheme, but we do not need to round because we were not asked. We simply were not asked to round to any number of significant figures. So just write your answer without any complications, 12.67. Describe how to determine the total mass of starch present in section B of this root. So this question is more determined about the cross multiplication idea. We need to get the total mass of starch present in section B. So section B has all these starch content masses in grams per 100 grams. So I need to get the total of everything, and this is corresponding to the total mass present in section B. So firstly, I have to get the total mass of section B. We need to cut out section B. I need to get section B all alone. So remove section A and remove section C and get section B alone in order to measure its mass. Determine the mass of all layers in section B, and after I determine them, I need to get the average starch content of section B. Average means that we have to add 31.79 to 33.26 to 29.64 and divide by 3 to get the average or the mean value, which will be a mean value of 31.556. Last step is use cross multiplication to get the total mass of starch present in section B. This would be by cross multiplication. The equation will be mass of section B times 31.556 divided by 100 grams. If I have fit this into a ratio, I would say that 31.556 are present in 100 grams of this sample. So if I have the total mass of section B, all of it, how much starch would we have? What's the total mass of starch? This will be my missing point. So if I need to get the x here, I'm going to use cross multiplication by multiplying mass of B times 31.556 and divide this by 100 grams to get the x. The x here is the missing part of this equation, which is the total mass of starch present in section B of this root. Last question, describe how this investigation could be extended to determine the variability in the concentration of starch. Where variability as a word, it means the variation of data using error bars. So in order to see that there is a significant difference or not. So in order to get this, I have to repeat the measurements in the same conditions. Take care that we have to keep the conditions the same. We need to calculate mean values in order to have a mean value in the graph. Then determine the standard deviation and plot error bars. So that's it guys for this video. I hope you found it useful and thank you guys for watching.